Emmy Bruns, from the Department of Biology at the University of Maryland. Dr. Bruns did her PhD research with Georgiana May at the University of Minnesota, and she was exploring the genetic and ecological constraints on the evolution of virulence in plant pathogens. And then started a postdoc to combine forces with Jonas Antonovics at University of Virginia to extend our knowledge of that model system of Dianthus, smut, and Silene and their interactions. She has widely published her research in the Journal Evolution and also the Journal of Ecology, as well as botanical journals such as the American Journal of Botany and the Proceedings of the Royal Society. In 2020, she took up a position as assistant professor at University of Maryland and in the Department of Biology. She's got greenhouse experiments going there. She's got field experiments at Beltsville. And she's been keeping a long-term study on alpine plants, which it looks like we're looking at right now in northern Italy. So she will speak to us tonight on the evolution of disease resistance in wild plant populations. Emmy?
All right, can you hear me?
Absolutely.
Okay, well, thank you guys so much. This is really fun to meet and know that there's a Washington Botanical Society. I actually had no idea, and your field trips sound really fun. That's great. Can I ask really quick, what time would you like to wrap this up? Because I'm looking now and it's 7:15. Do you want me to stop like 10 minutes before 8 for questions, or what's the, I have 3 little pieces and I might jump to the end piece earlier. So sorry, I can't.
It would be good for you to stop with time for questions and discussion.
All right, I might jump over one little piece then. Okay, so I was going to talk about a couple of things that we're doing here, and basically the driving question for me is, I think about disease a lot. I usually talk to disease groups. I tell myself as a disease ecologist, and disease, infectious disease, is ubiquitous in nature. One of the statistics I love is this one that there was a study done of this salt marsh in 2008 in Southern California, off the coast of Santa Barbara. And they found that the total biomass of parasites in this salt marsh was actually equal to the biomass of birds. So this isn't like numbers of species, this is just like total weight of trematode parasites is like equal to birds. And estimates of like how many parasites are out there, how many species is sort of all over the map. It's estimated that between 40 to 50% of all species on Earth are parasites. I feel like that's probably an underestimate because we're probably not counting all of the many viruses and bacteria that are less sampled than, say, the animal or protist parasites. So all of this really is sort of incredible. There's all this parasite diversity going on, all this disease, and yet the world is still green, of course, and plants manage to thrive and survive despite this sort of constant onslaught. And so I'm really interested in understanding, how do plant populations persist? They can't run, so how do they stand and fight? What kinds of defense mechanisms do they have? And how do they do this through time? How do they evolve mechanisms to avoid disease and to thrive in spite of disease? And so today I have a couple of things that I thought would be fun to share with this group. One is just give you a little bit of background into disease transmission in some wild carnations, and this is fun work up in the Alps I've been doing. It's late on a Tuesday night, so I talk about that, and then I'll talk a little bit about the evolution of resistance, and if I have time, I'll get into some stuff about the disease and the evolution of separate sexes I've done, which is more of a botany thing, which is a lot of fun. So briefly, I started working up in the Alps back in 2012 was the first time I ever came here. I started a postdoc and got invited to this just beautiful corner of the world. This is up in the westernmost corner of Italy here. This is the Mediterranean Alps. It's incredibly species diverse. The botany there is insane. And it's one of the centers of diversity for carnations. And this is the field site we work at. It's this little refugia. You have to hike up to it and you can stay there, and they have food and stuff. And it's lovely. There's a botanic garden right here. I'm trying to sell this as a destination for botanists. It's really excellent. And one of the other cool things, you can't see it in this picture, but there's this plant everywhere, Dianthus pavonius. This is an endemic plant called the alpine carnation, or the poppycock pink. And it's fascinating because it covers these slopes, bright pink blooms for about 2 to 3 weeks in the summer. And 43% of this plant is infected with this incurable sterilizing disease. So it's just everywhere. And you can see it right here in the center of this plant. And just to introduce you to the disease, it's called anther smut disease. It's caused by a fungus in the Basidiomycetes Microbotrium. There's a range of these fungi that do this. And quickly, sort of what happens, the life cycle, is we get these spores, these teliospores here, and they might land on a healthy plant. And the spores will germinate and infect the plant, and they'll grow asymptomatically inside the meristem. So you can't actually tell if a plant is infected when it's vegetative. It doesn't do anything outward to the leaves. It doesn't reduce mortality, anything like that. It's just asymptomatic. And then when the plant flowers again the following year, it has this symptom of smutty anthers. So on the anthers, instead of producing pollen, it's actually producing spores for the fungus. This obviously sterilizes the plant because it's not producing any pollen. And if it's, it also sterilizes the ovary. If it's hermaphrodite, that's sterilized as well. And it's perennial. So year after year that a plant gets infected, it will produce only these diseased, smutty, sterile flowers. And it's systemic. So it takes over usually the whole plant. So you can see these giant carnation plants that year after year are producing, 50, 100 flowers that are all covered with anther smut. And then we have that in a matrix with these healthy plants here. And of course, the transmission is really beautiful. And it's that these plants still look like flowers. They still produce nectar. And so they get visited by pollinators. And these pollinators pick up the spores instead of pollen. And when they visit healthy flowers, this stuff comes off onto the flowers. And so it gets transmitted. And so the same kinds of traits that might normally be good for a plant, cueing and attracting lots of pollinators, all of a sudden become a risk factor. And so the question actually that I started all this work with, and the question my colleagues and I were interested in, and just a little bit more since this is a botany group, this is a really cool fungus. And I'm going to talk primarily about 2 plants that get this fungus, but there's a whole suite of host plants that get anther smut fungi. They tend to all be in the Caryophyllaceae, and each plant pretty much has its own unique fungus. So there's some cross species transmission that can occur, but it's pretty rare. It's mostly sort of one host, one fungus. And so we see them in a bunch of the Silenes, Dianthus, Saponarias. It's pretty cool too, because it's actually preserved in herbariums. People would collect these things and not know it. So we have a beautiful historical record of where smuts were. And we also see them on iNaturalist. So like work that botanical groups go out, do, taking photos. We can use that to get at distributions of this fungus. And of course, there's some locals to watch out for. So Silene virginica, which is probably you've seen, it's not quite in this area, but further up, this one always has disease. You can most likely find it in here. And Silene caroliniana, I've been sort of desperately looking for the disease in the area and haven't found it. I found caroliniana. I know it's at Bear Island, but if anybody finds the smut on it, I would love to know. It would be fun to have a more local native plant to study this on. So anyways, what we were doing in Italy is this basic question, and all this work I'll talk about is with 2 collaborators, my postdoc advisor, Janis Antanovics, and another collaborator, Michael Hood at Amherst. And we were really interested in this basic question. Okay, this plant has this incurable sterilizing disease. Can this disease actually drive the population to extinction? And one of the reasons we think that a sterilizing disease could drive a population actually to extinction is it's not actually killing the plant, and so it's killing the next generation. So infected plants are staying in the population for their natural lifespans, which in this case can be up to 10 years, and continuing to put disease out. So it has all of the makings of something that could actually drive the population to extinction. And then we also had these data. And so this is some data that we'd collected. These were long-term studies where we'd sort of gone and looked at the same, measured the number of plants across a transect in the same place year after year. And we found in these 2 replicate transects in the area, here's the plant population size over time. Number of flowering plants is just tanking. And here's the average prevalence, the y-axis is over here, of the amount of disease. So it looks like, the plant population is crashing and the amount of disease is staying totally rock steady, the same. And so we thought, wow, this really looks like something where we could see disease-induced extinction, and maybe the disease is really having a big impact on when and where we find this host population in nature. But you can't really do, it's hard to project this out without knowing what the mechanism is. So we wanted to actually get into some population biology and try and model this properly. And so what we did was we actually did a marked plant study. So we followed the fate of 520 flowering plants for 6 years. We just marked these with like wire and went through with metal detectors year after year to find the same individual plants. And from this, we were able to estimate vital rates, things like transmission rate, how many times it flowered, mortality rate. And we built a model, and I won't go too much into this, but it was sort of a disease model where we were looking at sort of the transition from healthy to diseased and recruitment. And then we thought, well, first thing we'll do is we'll fit this model to our data, and then we'll show that the model works, and then we'll use this model to predict when extinction will occur. And I thought, we thought we were very clever. I particularly thought I was really clever, and it just didn't work at all. It was super crummy. And so what I'm showing you here is this is what we actually observe. This is the amount of disease we observe over that period of time, and all of these are runs of the model. So the model is just consistently predicting low disease. So we didn't know what was going on. We thought, well, how can we, measure these transmission rates and not be able to do this? And what we found basically was that in all this marked plant stuff, the transmission rate we actually observed was incredibly low. So only out of those 520 plants we looked at, only 16 of them healthy plants ever became infected, or 360. So it's about 4%. And that transmission rate is just not enough to keep the disease persisting. And we knew that it couldn't be spore dispersal that was limiting it, because you can actually go out and collect healthy flowers and rinse them off in alcohol and put them under a microscope slide, and they're covered in these anther smut spores. So they all, like the pollinators were doing their jobs, they were just moving these spores everywhere. So everything's trashed with spores. There's tons of disease in the area, but we can't actually see transmissions occurring in nature. And at the same time, we'd also been doing these lab studies where we grow up the little seedlings in petri dishes and inoculate them. And they were coming up like 80% infected. So we just didn't understand it. It was this mystery. They were super susceptible. The spores are everywhere. And yet we couldn't observe transmission in nature. And then sort of by accident, we found this result, which is that we kept getting really high when we would do these inoculation experiments in a dish with seedlings. We kept getting really, really high infection rates. And when I tried just by chance the same experiment, but with 1-year-old plants that were much more mature, they got infected at a much lower rate. And we've repeated this now, and this is actually data from the same experiment. And so what we found is that there's this incredible age basis to resistance. So the plants all start off very susceptible as seedlings, and they rapidly become really resistant as adults. And in fact, the infection rate of an adult, if you deliberately inoculate an adult, is about 3 to 4%. So the same infection rate we're seeing in the field. So we went back and we said, well, with this new information, well, so could there be, first one was like, maybe our model of all of the pollinators moving the spores around is not the best model for this system. We thought maybe there could be a second transmission route. This could basically occur if these spores, instead of just being moved around by pollinators, if some of them were actually falling on the ground just through a little bit of casual wind dispersal. And so we measured this just by putting out like transparency paper with sticky tape on it and pulling this up and then counting the number of spores. It was pretty high tech. And sure enough, you can find, the most spores fall right under it, but even almost a meter out, we're still finding a non-zero number of spores. So there are spores that are getting spread around, not by pollinators, but just by chance. In a dense population, there would be seedlings sort of in this mix. And so we think there's sort of 2 transmission routes here. And so finally, we put this new transmission route back into the model, and we sort of added in the fact that you could have 2 classes. You could have adult flowering plants and babies, and maybe the babies got infected by this higher rate, which we parameterized with our seedling inoculation experiments. And when we do this, we find that sure enough, I'm not showing you here, but the model fits much better. And moreover, we found that the only way that we can maintain the amount of disease that we see in the population is if almost 80% of the transmissions are occurring, not through pollinators moving to flowering adult plants, but to these sort of very tender little baby plants that are coming up nearby. So we actually think most of the transmission is to this seedling stage. And the disease actually really cannot persist without the seedling transmission. So if we take out these 2 arrows from the seedlings, we'd predict rapid loss of the disease over time. And it's really only can persist in these populations because of this high susceptibility of the seedlings. And so this sort of early work has really changed our idea of the basic biology of this fungus. So we now know that it has a mixed transmission mode. So we know that it's still transmitted, that the pollinators are still really important because it has this really wide distribution, and we find it in small populations where we wouldn't expect to find it if it was only this sort of very localized wind dispersal. But this aerial wind dispersal is really important for allowing it to exploit this extremely susceptible stage of the plant. And this exploitation allows it to persist in large populations, even when the adults have evolved high levels of resistance. And it's pretty epidemiologically important for this reason. So basically, this high susceptibility in the babies is allowing the disease to persist when we wouldn't see it otherwise. And so in my lab right now, most of our current research is focused on this question of, well, why are the seedlings so susceptible? I mean, this seems like a bad idea if you're a seedling, because if one of these plants gets infected with the smut, it hasn't flowered yet. And so the very first time it flowers, it'll be infected with this smut fungus, and it'll never produce any healthy pollen or any healthy seed. So it's just sterilized for life. So we thought, well, selection should be pretty strong against this. And so a lot of our work now has been trying to figure out this puzzle and ask, well, why is it that these plant populations are maintaining susceptibility at this juvenile stage? And for a lot of this work, we've turned to another system. So we're not working, we still work with the Italian carnation, but we also work with this guy who you may have seen, Silene latifolia. This is another European species, but it's been naturalized in the US. So it's sometimes it's described as an invasive species, but it's definitely a European species. It's a perennial, and both Silene latifolia and its endemic anther smut pathogen have been introduced here. So you can find diseased populations of Silene latifolia here. And we've switched to working on it for two reasons. One, it's got a faster germination time, so we can do a little bit more with it in the lab. And two, because it's naturalized here, we were able to get USDA permits to do field experiments and grow the plants outside and other things that make it a little bit easier. And so some of the questions we've really been pursuing are questions like, are these seedling and adult resistance, is this really two separate traits, or is it one trait? Like, can they evolve independently? Could you have selection drive up adult resistance and maintain seedling susceptibility? How independent are they? And how do these costs of resistance scale with age? And we think this is a pretty interesting question because, you know, if these traits are particularly costly, like if a plant, say, has lower reproduction because it's more resistant, that could prevent a trait from evolving. But we don't know how those traits are. Is juvenile resistance like a more costly trait than adult resistance? There have been very few studies that have looked at these. In fact, we can't find any that have looked at them in the same plant. So I'm just going to show you a couple of key results where we are with this. One is that we've found that there's really interestingly no correlation between resistance at the seedling and adult stages. So I'm going to show you this. We've actually done this in 3 species now. So along the so Dianthus pavonius, the carnation I showed you before, Silene latifolia, and then also another species of Silene vulgaris that's also sort of up in Italy and is also naturalized here. And here, the x-axis in all of these is the infection at the juvenile stage or at the seedling stage. And here's the infection at the adult stage. And so you can see in Silene latifolia, there's just no correlation. So things that are more susceptible as juveniles are not necessarily more susceptible as adults, and things that are quite as susceptible as adults are not necessarily more susceptible as juveniles. And then this is basically the same data, but each individual line here is an individual full SIB plant family. And so we think this is important because it's basically suggesting that since there's not a strong genetic correlation here, these things could actually be separate traits or have at least separate genes underlying them. And so we think they could evolve independently to resistance. And so we're interested right now, we've got a project in the lab where we're actually trying to do that with Silene latifolia, independently evolve seedling and adult resistance independently and see whether it drags along the other one or it leaves it, unaffected. Another thing we've found through our sort of digging around is that it's not just age that's really driving this change in resistance to anther smut, it's also stage or transition to flowering. So this is a former undergrad of mine, Liz Troy at University of Virginia, and she did this nice experiment with Silene latifolia where she used day length to generate different flowering times in plants that were the same age. And so here you can see that this is the proportion infected after we inoculated them. plants that were sort of in the flowering stage where they'd had a bolt shooting up and they were flowering were infected at a much, much lower rate than plants that were still in the vegetative rosette stage, even though these are all exactly the same age. She then repeated this, but instead of using day length to change the flowering stage, she used gibberellic acid. And it was the same thing here, where when she added gibberellic acid, the plants started bolting and flowering, and they got infected at a lower rate than the control, where we just added water. So it looks like it's both age and stage that are driving this. And it's really interesting because that all these plants are more resistant at the adult stage, because of course, once they're, or at the flowering stage, because once they're flowering, that's when they're actually getting visited by pollinators. And so we actually think this might be a response to selection, where you'd have stronger exposure to disease once you reach flowering, where you're encountering pollinators that are moving around, potentially carrying these smutty infectious spores. And then the last thing that we've been doing sort of on this project is this big effort where we've been trying to quantify costs of resistance at different ages. And we've actually found now significant costs of resistance at most ages, and I'll show you that. And we've been doing that out at the Beltsville experimental stage here in Maryland, as it said. So here's our common garden. We've been growing all of these Silene latifolia families. These are actually from just across the border in Virginia. And we've been measuring their fitness in the absence of disease. So we score the number of flowers and fruits and things like that. We've done it for and survival over 2 years. This is showing late May of year 2. They are really happy there. They do really well. And then these are all families that we've phenotyped in the lab for resistance, and we've quantified the resistance at 5 different ages. So we sort of know everything. And here's what we're finding so far. And I just want to give a shout out that I say we have done this, and here are the 2 people that actually have done absolutely the most work. This is Allison Keto, a tech in the lab, and Sam Swilinski, who's a postdoc in the lab. It's really been sort of a whole lab effort to go out and deal with this experiment. And so here's what we've been finding. And this is basically what you're looking at here is, in both cases, this is the total number of flowers produced in the 2nd year. And along the bottom here is within each graph is the percent infected following inoculation. So a high score here means very susceptible, because 90% of them are getting infected, and a low score here means very resistant. And then I've split it up. So we have male flowers in Silene latifolia that produce a lot more flowers, and then this bottom row is females. And then here you're going from age. So this is if we're measuring susceptibility at 9 days, and this is measuring susceptibility at 104 days. And so what we find is that, in fact, many of these lines, all of them except for this one right here and this one right here, are highly significant. And so we do find that there's this positive trend where things that are more susceptible are producing more flowers. Another way to think about this is, if you're more resistant at the seedling stage, you've got a lower average number of flowers than if you were more susceptible. And this cost seems to carry throughout the lifespan. We've also taken these data, because this is just sort of flower number in the 2nd year, and there's lots of other components of fitness besides that. So we've used this statistical program, Aster models, that sort of integrate survival and flowering over multiple years. This is useful for a perennial species into a single sort of integrative measure of fitness. And when we do that, we find the same thing, that basically there are costs of resistance in every single year, except for this 78-day one, where we don't know what's going on, and we're actually actively redoing this one right now to make sure it's not a fluke. But overall, it looks like there really are costs of resistance at all ages, but that they seem fairly similar, so that the slopes are quite congruous with each other. So we think that this is strong evidence that resistance is costly at all ages, and that these costs could actually be maybe partially explaining why seedlings are maintaining this higher level of susceptibility, and why you then have disease being maintained in populations even as adults become more resistant. So just some quick take home so far. I would say it looks at this point like anther smut is really unlikely to actually drive its host to extinction. And the main reason here is that the plants have evolved resistance, specifically strong resistance at the adult level prevents the disease from spreading very fast. In fact, the disease only persists in nature because of this high susceptibility at the seedling stage. And so far, we've shown that basically there is ample genetic variation for seedling resistance. So we do find some seedling families that are much more resistant, but they're not evolving to a high frequency in the population, even though we think that they're independent from adult resistance. And some of this could be that these are also costly traits to evolve. And so I have a quick little second story on these sexual morphs, but this is also a good stopping point, so I don't want to take up too much of your time. Do you guys want to hear about sex or finish and have questions?
I think we can hear the story.
You can hear about sex? Okay,
yeah.
This is also cool. So this is another little thing we've been working on, which is that a lot of these plants in the Caryophyllaceae are also known for their sort of sexual morph diversity. And so a lot of the carnations are what we call gynodioecious. So they've got hermaphrodites and females, and then diseased plants can actually infect both of them. And so if a diseased plant if this fungus infects a female, it just masculinizes it. So it causes the induction of anthers in the female plant and sterilizes the ovary. So it's pretty cool. It actually confused Darwin. When he was first looking at it, he thought it was N. Linnaeus. So there's a nice historical story about anther smut confusing people in the past, not realizing it was the fungus sex changing the plant. And so they can both get infected and the fungus masculinizes. And it's really interesting because one thing we've noticed is that there's a really high frequency of female plants where we find a lot of disease. And so this is sort of common across the Caryophyllaceae. It's a family that's really known for evolution of separate sexes. So we've also got complete separate sexes in Silene latifolia, true males and true females. And this has evolved independently at least two times within the carnation family. And we've got a lot of other examples of gynodioecious. And so the other thing we have in the Caryophyllaceae is a really high incidence of anther smut. And so I've been really interested in whether this could actually be driving this disease could be driving the evolution of separate sexes. And so in particular, what we're interested in is gynodioecious is often thought of as like a stepping stone to dioecious. And we're interested in whether smut might be sort of selection from this anther smut might be sort of moving plant species into having more divergent two different sexes as opposed to being hermaphrodites. And so one of the reasons for this is that basically as in a normal population, hermaphrodites would get half their fitness from pollen and half their fitness from seed. But as more females start to emerge in a gynodioecious population, hermaphrodites can actually gain more fitness out of pollen because there's more opportunity to pollinate too. And so that can actually then lead to the evolution of males. This is sort of the conceptual hand-wavy theory without any of the math attached to it right now. And so one of the reasons we think smut might be involved in this is in the hermaphrodite plants. They're what's called they go through male phase first, and so they produce their pollen first. After all the pollen dehisces, then the stigma develops and it's receptive. And then only after it's receptive, if it receives pollen, the flower closes up and starts to set seed. These female plants, as soon as they open, the stigma is receptive, and as soon as they get pollen, they close. And so what this means is that female plants are potentially just not open for as long. And remember, if pollinators are bringing spores around, there's probably a strong correlation between how long a plant is open and how many spores it gets on it. And so we thought that maybe female plants would have a lower exposure risk to the disease than hermaphrodite plants. And so we tested this up in the field. This was all really fun. This was all work I did with a bunch of undergrads from University of Virginia. And we tagged plants. So we bagged some of them so they couldn't get pollinated. We left some of them open. And then we looked at spore deposition on the open ones. We did this with females and hermaphrodites. And here's what we found. Basically, if you bag plants, then the hermaphrodites in the dark bars here stay open exactly the same length as females. But if you leave them open, then the females close much faster. This is the average number of days open, and the hermaphrodites stay open for longer, just as we thought, because they're dehissing pollen first, and then before they can get pollinated and close. And And we also found that on average, hermaphrodites flowers had a lot more spores on them than female flowers. So they are having a higher level of exposure in the field. And then I'll just say that we also know there's some interesting things I've showed you that, like, maybe you might be thinking, well, who cares? Because I've just shown you that these plants are really resistant. So if they get spores on their flowers, like, doesn't matter. They're not going to get infected. We've also found that there's direct fitness impacts of the spores. So we did these experiments where, and this was my undergrad, Audrey Betzel, where she was painting on either control, no pollen, just pollen, a pollen and spore combination, or just spores. And this is the amount of how quickly the flowers closed, the proportion that closed after 24 hours. But here, this is really interesting. When you pollinate flowers, this is the proportion that set fruit. And when you pollinate flowers and add spores, it drastically reduces their fruit set. And we've also shown now that it reduces their seed set as well. So just the addition of spores on the stigma doesn't necessarily infect the plant, but it prevents fertilization, and it like inhibits fertilization, and you get lowered seed set just from having these spores present. And we've shown this now in the field too. So we did this nice little experiment where we set up these pots of plants where we had a target female in each case, and a pollen donor, a hermaphrodite plant here. And then we either had 2 diseased plants nearby for like elevated amounts of spores in the area, or 2 female plants nearby, which would not be adding pollen or spores. And we did this, we replicated this. We did this over 2 years and replicated it. And I'll show you, this is the data from the one with the highest level of replication, but we found the same thing in a previous year that we had 9 replications. And here you can see basically, this is the average number of seeds per fruit in the target female. And these ones are the ones that had the female treatment. And then these ones here are the ones that had the disease treatment. And this is with a paired t-test. This came out as almost significant. And we've repeated this now and found the same trend. So what it looks like is over and over again, we're finding that the diseased treatment is actually having lower seed set. The plants that are next to diseased plants are having the females are having lower seed set than the ones that are not next to the diseased plants. So it looks like this is actually occurring not just when you sort of artificially paint things on, but also in a more realistic field setting. And then I'll just leave you with one final thing, which is that we asked, well, do diseased populations actually have more females? So this is what you might expect if the disease is actually driving the evolution of increased female frequency. And so to do this, this was a really fun field work. Here's the region of Italy that we work. This is the main population, Garelli, but we actually did all these different transects and sort of explored all these different areas. This was for a different project, but I sort of tacked this sex ratio thing on top of it. And so we'd go through and map all these populations, and within each population, we'd lay down a grid and count the number in a 100 square meter area, how many, or sorry, 10 by 10, how many plants there were, how many diseased, and what the sex ratio was. And what we found was that the sex ratio was dependent on elevation. So as you go up in elevation, you get more females, but it was also dependent on the disease status of the population. So in blue, and this is female frequency here. So in blue and this solid line, this is populations that were that had disease in them, plant populations that had disease, and this is the female frequency of those populations. And then this lower line here is the population of females that in populations that were completely healthy. And so it does look like females are more common in populations that have disease, which is supportive evidence for this idea that disease might be driving the evolution of higher female frequencies. And I think that with that, I have a little section on, I had some stuff about pollinators, but I'm going to jump ahead and just say, basically through this, we've shown that females have reduced exposure to spores, both through closing behavior and through pollinator avoidance. And females are more common in diseased populations. And we think actually there could be 2 explanations for this. So one might be evolutionary. It could be that disease is selected for female bias sex ratios. And the other one could be an ecological one. It could be that disease is actually spreads more easily in female biased populations if pollinator behavior changes such that vectors are actually less satisfied with the females and moving around more, which we also have a little bit of evidence for. And so I'll just end by saying thank you to a bunch of people that have helped with this work over the years. We've got a couple of collaborators in Italy that have been totally instrumental to this. Valentina Carasso at the seed bank that's run out of the park that I work at there, and Ivan Pace, who's the lead botanist out there. A bunch of different undergrads have contributed to this, Sam and Allie, and just a ton of, we're never short of people that volunteer to come and help with the field work. And thank you to you. And so I'm happy to take any questions.
Thank you very much. That was really interesting and tremendously comprehensive. There are questions in the chat. We could maybe start there, and then.
I'm so sorry, I didn't see those if those came up earlier.
Can you see those, or do you want?
Oh, yeah. Gosh, okay.
Okay.
Oh yeah, it also occurs in the Montiaceae of the Andean Chile. What is the seed set ratio in the species? Sorry, I'm not quite following all this.
Is Rob, are you there? Do you want to explain your question a bit more? Okay. Well, let's move on to the next one. You can see there's a question here about plants that survive on without fungus, and they
have genes for resistance contrary to those seedlings that did the plant. So that's a good question. We're working right now to try and understand what the genes are that are actually underlying resistance. One of the tricky things there has been to try and we can cross for resistance, but it's we always have to screen for it by infecting everything within a family and then looking at the proportion that get infected. And so we don't know whether we're still seeing segregation coming out of that. But right now we've also found a way to breed for susceptibility, which it turns out, and I think this answers another question too about climate or season, which is that we've found that at really high temperatures, you can end up with a temporary curing of the smut fungus. So at really high temperatures, and we're seeing this especially in Maryland now because we're growing Silene latifolia plants that we collected from the Piedmont down in the flatlands here where it gets really hot, and in the heat of the summer, they lose the smut temporarily and they can produce healthy flowers and cross them. And so we don't know what the genes are yet that underlie resistance, but we're actively looking for it right now. So we've been selecting for resistance on the one hand at both adult and seedling stages, and then also susceptibility by growing things, selecting ones that get smutty, and then curing them and crossing them. We don't know yet.
Yeah, maybe you can hear me now.
Yeah.
I was wondering what the average seed set is in uninfected populations versus populations that are infected.
That's a great question, and I don't have a great answer. Getting that average seed set has been really difficult, and we only have sort of average seed set data for one of the main populations that's pretty infected, and so I don't know the seed set data for a healthy population, in part because I'm usually there for the flowering season and don't always stay into the fruiting season. And the other part of it is that we think actually that the seed set is super variable season to season anyways, because there's another player in all of this, which is that they also get a moth, a hadinid moth that comes in and both pollinates, but also sometimes lays eggs in the flowers. And sometimes when you go to collect seed set, there will be populations that have just gotten hit and you can't find any seeds at all from them.
Yeah, well, I mentioned that because in outcrossing plants, Michael Bazaz has found like an average of 20% seed set versus like inbreeders that can set lots of seed, or apomix that can set lots of seed. thrips and things and other insects can get in and, of course, really mess up the seed set for any particular population or season.
Yeah.
And
Oh, we have
like 0 to 10% in dioecious,
mm-hmm.
dioecious grass and of seed set compared to things that are inbreeders. I said 80, 90, 70% seed set. So I'm curious what the seed set is in the species you're investigating. Well, I guess
we've looked at, so we haven't looked at seed set in the field so much, but now that I think about it, we have looked at seed set of families from different populations, and we've actually looked at we've looked for evidence of inbreeding depression in those families, and it varies quite a bit. Oh, I even have a slide here. So we've these were populations that Gorelli is the main area that has a ton of disease, and then these other areas they also have disease, but not quite as much. And we were looking at sort of deliberate crosses within population and then with self, and we found that there was some pretty strong inbreeding depression in some of the populations in terms of like strongly reduced seed set, and in others not so much. We found no difference in seed set between females and hermaphrodites. So a lot of the common thought about gynodioecious species is that females just produce more seeds, and that's the advantage. But we haven't found any evidence for that. But I don't have great sort of field data of seed set across lots of different populations. Be great to have.
Okay.
So there's questions about does the plants that survive without fungi, about whether infected seeds are transmitting this.
Oh yeah. So do infected seeds germinate and carry the fungus? So we don't know the exact stage when the infection occurs. We don't think that the fungus can last very long in the soil. So the spores, the fungus itself has to pretty much be inside of a plant to survive. It has a brief period where it can live as a yeast on the surface of the plant or in it, but it could be going onto the soil, and then if a seed came up right away, germinate and infect it. But we don't think it's like getting in through the seed coat or anything like that. And it's certainly not, you can't have like a plant that produces diseased seeds, because an infected plant is sterile and not producing seeds. But very young plants that germinate, and if they were like grow up out of a lawn, with spores all around, they would probably catch that, get infected with the fungus right then. Is there evidence of apomixis at higher elevations? I don't know. Not that we've seen. We find that there's more females at higher elevations, but we haven't found any evidence of apomixis. There's probably some cryptic stuff going on where some of the hermaphrodites maybe have lost their ability to set seed, but there's nothing morphological in that. So we don't have a good sense of how far up that goes either. Does climate or seasonal weather have an effect on the number of plants that exhibit the smutted form? Yeah, so actually we think that climate is probably having a pretty big effect here. As I said, it gets likely not in these high alpine carnations. There I found that in growth chambers, I have to jack the heat up to, almost like 32 degrees Celsius or something to cure them. And then the plants barely, the plants might lose the fungus, but they're so crummy looking that they can't really reproduce either. So their heat's bad for everybody. But at lower elevation, some of these Silene species really, under high heat, they do lose the disease for a couple of periods during the summer. And we're actually really interested in that right now, because that sort of period of summer, and the plants seem to do fine. They can still flower and produce seeds under that high heat. And so we're interested whether increasing in climate change might reduce the overall distribution of the anther smut fungus, and also whether it would sort of change selection for resistance, and whether we might see sort of increased susceptibility if plants just aren't facing the same kind of disease pressure that they would under cooler climates. Oh, and I would be delighted to check the incidence of smut in the 2 sites at Marymount University. That would be great. Yeah.
Yeah, I'm happy to help you with it. I'm really excited.
Yeah.
We did something like this with the USDA and a downy mildew, and everything was disgustingly healthy.
Okay.
Maybe we'll find something. So we can email, later on.
Yeah, no, it would be great. I sort of troll iNaturalist occasionally looking for this, and my colleague Michael Hood has actually, published a few papers using iNaturalist data to try and look at distributions of it. But yeah, we probably miss a lot. People don't, and yeah, it would be great to know some more local sites. Mm-hmm.
Amy, I have a question about nectar production.
Yeah.
So, I mean, you've measured so many things. I can imagine you're like, maybe that's not going to be something we do. But I mean, are you seeing any trends and variability of nectar?
Yeah, so we did measure that, and we've found that diseased plants make less nectar overall than healthy plants of both sexes. And in fact, we've also looked at pollinator preference for the two, and pollinators do tend to avoid diseased plants, and they preference healthy plants, but there's no significant difference in the females versus the hermaphrodites. So those seem to be making about the same amount of nectar.
And just a real quick other, the sort of stigma clogging with the spores is so cool. And you don't suppose there's any sort of chemical, interactions or anything like that going on, allelopathy or anything like that?
So it definitely could be, and we're not the first people to have found this. So we actually looked for it because it had been reported in some other anther smut systems, and I think we're interested in trying to connect that function to okay, how important is it actually in a field setting? But I know that other colleagues have tried. You get the same effect if you use live spores versus heat-killed spores. So it's something just about putting the spores on the stigma, and then, but it's a different effect from you don't get the same effect if you put con-specific pollen on or something that might also sort of cause a non-self recognition. So it definitely seems to be something about the spores, but whether it's chemical or some live thing, it's unclear. Other than the heat-killed ones do it too. And there's also a dose response. So if you do very small levels of spores, you don't get that. But if you sort of paint them on kind of thick, you do get that. And we've actually gone back and looked and tried to take the, I didn't show this data, but you can quantify the amount of spores it takes to cause that reduction in seed set. And then we've done a bunch of stuff where we just count stuff all the time. So we were just counting spores. I tell my students, it's okay, you're an ecologist, you go and count stuff. That's all we do.
Right.
So we're counting spores on flowers. And so we know that there's, most flowers have some spores, but some flowers have a lot of spores. So those are probably plants that got a direct hit. And those high levels of spores, it's 7,000 spores or more or something. Those are the ones that we think is a high enough dose to actually reduce seed set in that flower. And so we were trying to calculate out, well, okay, if you had this number of flowers that were infected with this, what would the actual impact on the population be? And it's not insignificant. It's, it'd be like a 10% reduction in overall seed set.
I think there's another question in the chat. Got a second more here.
Oh, is there a seasonal timing variation in expression of infections? Oh, and also Bear Island, you'll see Silene virginica. Would you really see virginica at Bear Island? Is it the bright red one? Or is that Silene caroliniana? I know I found caroliniana out there, but I've never found it diseased.
Diana.
I think it's caroliniana, isn't it?
Yeah.
I think that one's a really interesting one. It's a much longer lived species than virginica, and we find it diseased, but it's rarely diseased. And actually the Silenes in North America, they have really different anther smuts than the ones in Europe. So it's like a deep evolutionary branch in the microbotriums there. It's pretty interesting. And yeah, is there a seasonal timing variation in expression of infections? Yeah, we think there is. And one of the things that's really interesting is that the disease, when it gets into a plant, can change the phenology of the plant. So infected plants tend to flower earlier than healthy plants in almost all species. And actually, this is work not so much that my lab has done, but my colleague Michael Hood has done a lot of this using iNaturalist data, because so many people collect data on plants that you actually get those beautiful phenology curves. And then you can go through and score which ones are diseased. And you can see that in most species, the diseased plants are flowering on the early end. And there's a couple of species where they're flowering on the later end. And there was a study recently of looking at some of these carnations in the Himalayas. And they found that there were 4 of these species where the diseased plants flowered way, way, way earlier than any of the healthy plants, which doesn't actually make a lot of sense, right? Because if you're a diseased plant and there's no healthy plants flowering, like, how are you going to infect new plants? And so we actually think that some of these flowering phenology adaptations might actually be adaptations for infecting seedlings. So they might be lining up better with when the seedlings are germinated to try and hit that young seedling stage than the flowers, which is what we always assumed. But it seems like so much of the infection biology is at this sort of cryptic young stage rather than the one that you can like observe and watch the bees and the butterflies move around in between.
All right, so it is, I think, a little past 8. So do we have any other quick questions for Emmy?
Hi, everyone. I have a quick question. Yeah, nice talk.
Thanks.
So my question is, you mentioned other species and genera on Caryophyllaceae have, they also exhibit the fungus, right?
Yeah.
So my question is there any, have you seen a phylogenetic signal on like on the fungus and on the genera or species parasitized by the fungus, like all of them are in a single plate within Caryophyllaceae, or do you have any phylogenetic specificity?
Yeah, there's quite a bit of phylogenetic specificity, and it's actually, I generalize, it's not just the Caryophyllaceae. There's been the most divergence in the Caryophyllaceae. If you actually look at the phylogeny of Microbotrium, the clade is older, and the sort of the basal species in the Microbotrium clade are infecting individuals in the Polygonaceae. And there they don't cause an anther smut, but they cause like a floral or an ovary smut. So it just sort of like blows up the whole flower and it's wind dispersed. And there's some other species as well that are sort of more flower or ovary type smuts that are in other plant families. So it's not just confined to the Caryophyllaceae. The Caryophyllaceae is just best known because it's really radiated within that group and caused a lot of these more characteristic true anther smuts where it's infecting the anthers and not sort of disrupting the whole flower. We've actually seen one reversion within the Caryophyllaceae going from an anther smut back to a floral smut and back to wind dispersal. And we think some of this like seedling adult resistance is maybe important in that. But we also know that the phylogenies of the host and the pathogen are totally not congruent. So it's a history of like occasional host shifts and then adaptation to a new host, sometimes from a close phylogenetic related, but sometimes the shifts are a bit more deep. I don't know if that answers.
Yeah, I'm asking because many members of Caryophyllaceae are dioecious, including in Polygonaceae and other groups. So I was wondering if maybe some of them have developed resistance or a way to minimize the incidence of that form.
Yeah, it's they're definitely less. I mean, when you go out looking for these smuts on things outside the Caryophyllaceae, they're harder to find. So the other thing about a lot of the species in the Caryophyllaceae, not only do a lot of species have them, but you can get to really high load, like disease loads or disease prevalences, suggesting that maybe they have lower overall resistance. Maybe these other species have evolved higher. It's hard to study disease in species that don't have a lot of disease because but probably important too, right? Because maybe we're missing something important like resistance mechanisms, old resistance mechanisms that have evolved.
Thank you. Thank you so much.
Yeah.
Well, I think we should all thank Dr. Bruns for a really terrific talk with so much to think about. And it seems like you have an endless system for exploration. It's pretty exciting. I guess I can tell you what our next talk will be on March 7. We're going to have a talk by Dr. Karen Gadan from the Department of Biology at George Washington University, and she'll be talking about climate change and marsh migration. She's been studying marshes in our local area, and she'll talk about that. Anything else, Gabe? Do you have anything to add?
No, not at all.
All right, so I again, thank you so much, Dr. Bruns. It was terrific, and please come join the society.
Oh yeah, no, this seems
terrific.
great. Thank you.